No screeching. Okay. Oh no. Oh shit, it's so slow.
Mine?
Yeah.
Yeah, see, that's what I'm saying.
Okay.
Like, mine's gonna be a problem.
It's
I don't know
okay.
why.
Mine would also be a problem. Okay, cool. All right. Welcome, everybody. Kind of a big deal. This is Yago and I's last ever reception that we're holding. Oh, I know we're crying. I know we're so sad. Oh yeah, thank you, thank you. Some claps, maybe. Yago and I are both seniors, so you guys will still have review sessions after this, but it's Yago and I's last one, so like a little sad, kind of excited, but mostly sad. If you just joined, we're doing topics 4 through 7 for your exam on Thursday. Just some general thoughts, like how are we feeling compared to exam 1? How are we feeling? Better than exam 1? Okay, thumbs up for like better than exam 1. Okay, this for like kind of I don't really know. Okay, and then thumbs down for like okay. All right, cool. Good to see that at least most people are like halfway here. We're gonna go over some questions. We do have like a lot of content, but we're going to go through that pretty quickly to do some more question stuff. If you have a question at any time, just raise your hand. But if we don't call on you right away, we're just like trying to finish up a slide or something. But feel free to ask questions at any time. Anybody have anything super specific that they want to go over in this review session?
Uh, it's about like looking for like historical start sites in the DNA strands.
Okay, with like, are you thinking about like the quiz 4 question that was like the start sites?
Yeah, or like the problem set.
Okay, yeah. If you remind me, we can go over how to find those. Anything else? Any specifics? Question mark, any specific problems you remember from the quizzes or anything that really rubbed you the wrong way, other than the potential start site question? Okay, if it comes to you, just let us know. But the goal of this is just like get you a little bit more confident about taking on Thursday. Good news is we've already done this before. You know what it's like. And this is going to be, really claim your happiness now before the final, because the final's going to be tough. But really, this is yay. This is like best part of the semester. Yay, exam 2. You're gonna miss the days of exam 2. Okay. All right. Okay, we're gonna start with mechanisms translation. Nago's gonna start for us.
Is this gonna be more conceptual or is it gonna be nitty-gritty?
No, this is gonna be so the content is basically big idea stuff because we have so many concepts to talk about. But if anyone has little nitty-gritty questions, feel free to ask them. And we
Which is all open. Same way as exam 1.
Same thing. Same thing.
All right, so first topic we're going to go over today, topic 4, mechanisms of translation. All right, like we've covered before, translation is just basically that process of the mRNA copy being turned into the protein, right? So we started with transcription. The DNA was translated into the mRNA, and now we're using that mRNA code to make the protein. That the translating is done by tRNA and rRNA in the ribosome. So the ribosome has ribosomal RNA, which is the RNA part, and it has the tRNAs, which we'll talk about more, that bring in the amino acids onto the code and create that polypeptide chain. All right, prokaryotic translation. This is broken up into 3 parts. First, we have initiation, which is where the ribosome binds on the mRNA. First, the small subunit, which is like the bottom. The ribosome's like a hamburger buns, I guess. So it'll bind first to the small subunit, then tRNA is going to bind at the start codon, and then the large part of the ribosome binds. So this will be a little different in the eukaryotic transcription. So that's important to know. First, small subunit binds, then tRNA, then large subunit. All right, that's how you start an initiation. And then after that, you have elongation, which is when new tRNAs come and base pair to that mRNA, and they're matching codons, and each of those tRNAs adds a new amino acid. So each of those amino acids like glycine, alanine, all that, will be brought by one tRNA. And then each tRNA just builds up a whole chain eventually of them. All right, as we have the chain being made, once we have the special amount of amino acids that we need, is termination, and that's when the ribosome reaches the stop codon and it releases the mRNA and the chain that it had made up. So after termination, now the ribosome is released, and you have just the separate mRNA and the separate polypeptide chain. Peptide bonds, covalent peptide bonds.
And quick question, what is the name of the amino acid that the stop codon codes for? None. Right. Okay. Does anyone remember the name of the start one? Yeah, it's like just like met. I just like think of it as met. Yeah. Yeah, I think you should. What was it last time on the quizzes? Has she given it to you on the quizzes? Yeah, okay. Okay, if she's given it to you on the quizzes, then I would say yes. But really remember, guys, like she's gonna hit you with questions like how many amino acids, and we're gonna have a question like that later. How many amino acids or blah blah blah. And you have to remember that the stop codon does not code for an amino acid. All it codes for is just stopping it. The start one codes for an amino acid, which is the MET, METs. I just think MET in my brain because I don't think of the whole word, but just remember that. That's important.
Do you hear about release factors for termination?
No.
Okay, so this is basically an image showing just what we talked about. We'll have the initiation right here. So the tRNA is coming, binding. This is the tRNA right here. It'll have the anti-codon, which is a three-letter codon that's complementary to the codon it wants to bind to. And it brings it in, binds, and it has an amino acid, a specific amino acid for each codon. And so that'll bind, and then it'll move over here, and here is where the elongation step happens. So you can see that the amino acids are being added on. This one would have been from the previous tRNA, this one from the one before that, and so on. And then as soon as it does that and attaches, it's leaving, and a new one's coming in. And that keeps happening until this chain is full length. And once it reaches that stop codon, the chain's full length, the chain comes off, and the mRNA comes off. And then this whole thing is the ribosome where this is all happening. Like we talked about, it's like a hamburger bun. We have the small subunit at the bottom and the large subunit on the top. Does anyone remember the order of the binding that we talked about in the last slide for this ribosome on the mRNA for prokaryotes?
Talk about which subunit?
Like like the order of the binding that I talked about in that. So it'll be the mRNA binds to the small subunit right here. Then the tRNA comes in, and then the large subunit. Okay? Does everyone understand that so far? Yeah, so how I think of it is the first is the A site. So I think attachment. This is where it's attaching, right? And then P, you can think peptide or prolong. All right. So that's elongation. The chain's getting longer. And then E is the last one. And I think exit. What's up? Since it's tRNA, I would assume U because it's RNA.
For anyone who doesn't remember, anticodon is what, if you look on the tRNA, it's like the bases, the groups of three that are attached to the tRNA. So that's how they know which codon on the mRNA to bind with. That's what the anticodon is. Yeah, sorry.
When the large subunit binds on the tRNA, does it put it in the P site or the A site first?
It would still be binding when it's in the A site, and then it would go to the next one.
So mRNA to bottom of the hamburger bun, that's the first. tRNA comes in, binds, and then top of the hamburger bun.
So it binds in the A site?
The tRNA binds in the A site. The top part of the ribosome just binds like over the whole thing. Because the top part of the ribosome is like where the part catalyzing the peptide bond formation.
It's that for both prokaryotes and eukaryotes, the initiator methionine tRNA is placed or binds to the mRNA and the large ribosomal subunit binds on top, placing the tRNA in the P site.
We'll talk about the
That-
differences in a second.
That's for eukaryotes.
Yeah, we'll go over the difference. We have them like the next slide, pretty much. This is for prokaryotes.
Okay. Eukaryotes, same idea, but the differences are all in initiation. So in that first step. So instead of the prokaryotes, like we talked about, where it's the mRNA binding to the bottom small subunit of the ribosome, then the tRNA binding, then the top subunit. In this one, we have the tRNA binds to the small subunit first, and then all that binds to the mRNA. And here, instead of binding at the start codon, it's going to bind at the 5' cap. Does that make sense to everyone?
Here, so let's go over it one more time. So somebody remind me for prokaryotic translation, what's the binding order? So it's mRNA binds to the bottom hamburger bun. That's number 1, right? Number 2 is what? Somebody remind me. tRNA binds to what?
Start codon.
Start codon, right? That's step 2. So step 1, mRNA, bottom of hamburger bun. Step 2, tRNA binds to that mRNA. And then what's that third step? What's the last part we're missing? Yeah, large subunit, top of the hamburger bun. So that's prokaryotes, right? Now, eukaryotes. Let's go over the difference just one more time here so we all get it. So usually you have the mRNA binding to that bottom subunit first, right? That's what we know in prokaryotes. This time, it's the tRNA binding to that bottom of the hamburger bun. That's now step 1 for eukaryotes. Okay, that's the difference there. And then binds to the mRNA. So do you see the difference in steps there? So again, prokaryotic, it's mRNA to bottom of the hamburger bun. Second step, tRNA to the mRNA, and then that last hamburger bun going on top. Eukaryotes, that tRNA binds to that bottom hamburger bun. That's step 1. Step 2, binds to that mRNA, and then we'll have that last guy. Does that make sense? Difference in steps there. Really think about it in terms of steps. Okay, and use what Yago has some good like memorization techniques for all of this. So A was what?
Attach.
A was attach, P was prolong, E exit. Got it? So you're moving, starting here, attaching here, moving it, we're shifting over. And what are those bonds that connect all the amino acids? What are those called? Peptide, right? They are covalent bonds. They are covalent, but they're peptide bonds, right? So when we're talking about the string, that's really important, right? So we're making a string. We're starting at A, going to P, ending at E, and we're adding more amino acids to the string. That's all we're doing. Does that make sense?
And think it's a polypeptide, multiple peptide, so peptide bond. Does anyone know what makes the peptide bonds? Yeah, but think what she would want you to know and say for that is ribosomal RNA. So our RNA in the ribosome. All right, and then the rest of the steps, similar, basically the same to what we talked about for prokaryotes. There's the elongation at the P site where the tRNA is moving to the P site. Its amino acid is being added onto the chain. And then we have termination where it goes to that E site, the exit site, and it just leaves, the mRNA leaves and the tRNA leaves, and then you will have the free polypeptide chain.
Yeah, that whole like long phrase, like catalyzes the hydrolysis of bond between, that just means that they're cutting the bond between that tRNA and the chain that we just made. That's all that means.
All right, so then we have point mutations. Point mutations are just a change in a single nucleotide. So think point, 1 point. There's a bunch of different types of point mutations you can have. You can have a silent point mutation, a missense point mutation, nonsense, and a frame shift. All right, these are kind of descriptions of each and images. We'll talk about each more in depth, but this is a helpful picture to study them all. if you guys want to save that or look at it later.
And real quick, can somebody explain to me what we mean when we say we have a frame? What does that mean? What does that mean? I know you know it. Anyone else? Anyone else? Yeah. Right. Okay. So you have long mRNA, right? Long mRNA that needs to be made into amino acids, right? How many nucleotides are in a codon? Three, right? One codon makes one amino acid, right? So when we're talking about frame, what sets the frame? Does anyone know the answer to that? What sets the frame? Yeah. Start codon, right? So I could have like the longest string ever, right? We're talking about mRNA, so it's like super, super long. Until I get to that start codon, I'm not starting anything, right? I don't care what comes before the AUG. AUG happens, and when we say we set the frame, AUG, that group of three, sets the next groups of threes, right? Because we're starting there. That's what we mean when we say frame. So when we add a nucleotide to this string or we delete one, that whole frame is shifted because now it's not the same groups of three anymore, right? We're shifting that frame. Does that make sense to everybody? I know that frame thing kind of confuses people. Yeah.
All right, I thought there was going to be more slides going into each specifically, but there's not, so we'll just cover them here. For a silent point mutation, silent just means that there's a mutation, but you don't notice the difference because the amino acid doesn't change at all. So it's like it didn't even happen. You can see here, this is the original code, and then here we have the change. So we went from a T to a C. But if you look at that chart you guys have of what codons code for what amino acids, TAT is tyrosine, and so is TAC. So we changed the T from the T to the C, but you can't even tell because tyrosine is shown up for both. All right, so that's a silent mutation. Then missense mutation would be a change in a nucleotide that changes the amino acid. So this one we actually see. So we have TGG here, that was coding for tryptophan, and now it changed to TGT. So it was one codon change or 1 point change, but now it's actually changing to cysteine from tryptophan. So that would be a missense mutation. Now we have nonsense. Nonsense is just a stop. So we changed one of the letters, and it happened to make it a stop codon now. Okay. So missense and nonsense are really similar in that they changed just one and they changed the amino acid, but the difference is just missense changes it to an actual amino acid, nonsense would change it to a stop. Does that make sense? Those are hard to remember, easy to confuse on exams. So make sure, even though you think you get it, keep going over that and the difference between the two. And then last one, like Anjali talked about, frame shift. Think frames are just groups of three. If you're adding one or deleting one, it's going to set off that order of threes because now you're going to have an extra. Yeah. Point mutation is just a change in a single nucleotide. So each of these letters is a nucleotide, right? That's the base for a nucleotide. A point mutation would be changing one of those nucleotides. So you can swap it with another one, or you can take it away, or you can add an extra, but it's just ones. Frame shift.
These are all point mutations.
Frame shift.
Everything on this slide is a point mutation. I would say that would be a missense, just because, like, again, the start codon does like code for something, right? It does code for that amino acid. So if we change it and it changes it to a different one, then yeah, that would be, even though that's not ideal because we need that start codon, if it's still changed. So that would still be a sense. You want to answer?
It changes it to a stop codon in general, but most of the time it causes it to come earlier than what it was supposed to. Because this could be the DNA. Yeah, I don't know. I mean, this could be they're just using these as examples. I don't know if each of these codons actually codes for those amino acids, but just the main idea is that it would code for a stop instead of what it was coding for before. Makes sense.
All right, question. Okay, how many amino acids long would the protein from this mRNA sequence be? And what is the amino acid sequence? We'll give you guys about like a minute to do this on your own. Really helps to write it out. Sometimes on the exam, when I took this class, I like to like put little brackets around each three so I know where the frame is. That makes it easier. But again, also notice with the chart here, multiple things can code for the same amino acid, right? Let's keep that in mind for when we're talking about missense and we're talking about nonsense mutations and silent. Multiple things, multiple different combinations can code for the same amino acid.
Just do it. Just finish that if you're good with the next two.
Oh, wait, it's actually not. Yeah, okay. I'll just like jump in. Okay, all right, let's start. Anyone know? Anyone just tell me the number of amino acids long you think it is. Yeah.
4.
4. Okay. And how did you find out that it was 4? What was the process there?
I looked at for the start codon and then I looked for a stop codon,
Right. And what is the start codon? What is it?
AUG,
AUG, right? And which direction do you look for it from?
from 5 to 3.
5 to 3, right? So number one step, finding that start codon, 5 to 3 AUGs. That's what you're looking for. All right. I see one right here. It's about like, it's pretty early on. I highlight that. That's my start codon. That's my green. And then if you look on the side here, we have a couple different opportunities for the stop codon. What is the stop codon? UAA. In this case, it's UAA. Okay. You can go to the next one. Actually, no,
One thing, when you're looking for the start codon or the stop codon, so let's say you're looking for start, so AUG, can you just look for AUG anywhere, or do you have to watch out for something? Yeah. So what about DNA? Like think the frames we were talking about. You have the 3, right? So if you have, so here it works out because we have 3 and then 3, right? So these are all in the AUGs in the same group of 3. But say it was just GG. So no, say it was, we get rid of the first G, right? So ignore this G. And now it's the first 3 would be GCA. So that A wouldn't be part of the UG anymore. Does that make sense? So be careful. We want to look for those start and stop codons, but we want to make sure they're in the frames. Okay. For DNA though, like because it could shift like the amino acid coding for it. Right? That makes sense. Like say you have the tRNA coming. The tRNA is coming, right? Well, for the DNA. The DNA is making the mRNA. The tRNA would bind to the first like codon or the first frame. And then it wouldn't match. Does that make sense? Basically, you just want to make sure that the 3s, like it's part of the 3. You'd want to make sure it's not a separate one.
And that would be
For this and for any for any amino acid, so not just the start. Make sense?
Okay, does anyone know what the 4 amino acids in between, what do they code for?
So if we know it starts here,
What's the start one? Thymine. Who can name the next one? Just the next one. Proline. Third one? Yep, arginine. And what's that last one?
Valine? Yeah.
No. Glycine, right. And what does the stop one code for?
No.
Nothing. Nothing, right?
here's what we talked about. We have the start at the AUG, then we have the stop UAA, and then in between. So this will be 1 amino acid, another amino acid, third, fourth, and then the stop isn't one because like we talked about, stop doesn't code for any amino acid. Exactly, yeah. So say this GGG, instead of the G here, the U started there. So if we were looking at it, we would see a UAA, which we would think is a stop codon, but that U would have been part of the GU. Make sense? So then it wouldn't have been a stop codon. So like we know the start codon's there and we're counting by 3s after it, but that one wouldn't have been in the same frame. Yeah, exactly.
And just remember that start codon is what sets the frame. Right? So talking about like the stop codon here, the AUG sets the frame for everything after it. Okay, when we're talking about amino acids, because AUG is the starting one, and then we care about all the amino acids. That's what's going to be made, right? So just like Yago said, if we have the same AUG where it is, and we take out that G right before the UAA, that frame is now shifted, right? Because it was set by that AUG, and now I'm going in groups of 3. Does that make sense? So again, just think about that AUG. That sets the frame for everything after. Any other questions?
So even if the frame shift is in the start codon, it's still,
No, just think about it for after the start codon.
So I don't care about anything until I see that start codon. Until I see that AUG, that's what sets the frame. Everything after that, I need to look at it in groups of 3, right? Because I'm starting with that MET, and then I need to look at the next 3, which is whatever the next amino acid is, blah, blah, blah. So again, if I like take out something after the start codon, before that stop codon, if I take something out, or if I add something, or if I change something, you just need to look at it in groups of 3. So AUG sets the frame for the rest of it, right? And just like Yago said, if instead of that G, if I took out that G right before the UAA, and I still looking at it in groups of 3, that frame is now shifted, right? Because we've messed with stuff after the AUG. And the AUG sets the frame. So she's had this on exams before, like she's been like, explain what I mean by the frame and what sets the frame and blah, blah, blah. What sets the frame? What is it? Start codon. Okay, start codon sets the frame. When I say frame, I mean how we look at it in groups of 3, because 3 nucleotides codes for a codon, and each codon codes for a what? Amino acid. Right? Does that make more sense? So AUG sets those groups of 3. That's it. Okay.
Yeah, we're talking about here the mRNA.
Do you have questions? Do you want me to re-explain anything, anybody? Okay.
All right. What are the different types of RNA used in translation, and what does each do?
Somebody name one. Name one. RNA.
So rRNA would be what? What does the R stand for? What's another name that they'll call it? Ribosomal RNA. Where would we find it? In the ribosome. So it makes up part of the ribosome. And what does it do? It's what catalyzes the peptide bond formation, right? And the peptide bonds, like we talked about, are the bonds between the different amino acids that make that chain.
Let's string it together. That's the peptide bonds. So without RNA in that ribosome that makes up the ribosome, we can't make that string.
All right. I'm thinking 2 more types of RNA. mRNA. What does mRNA do? Yeah, so the mRNA is the part is like the code from the DNA, and we're using that code in translation to make the amino acid strand. And last one, tRNA. That's does what? Yeah, exactly. So that's what's gonna bring over the new amino acids by binding to the complementary codon. Here's what we talked about, the mRNA, that's the code that's being translated by the ribosome. Then we have the tRNA. Those are like those little loop things binding to that mRNA, and each one has a certain amino acid at the top that it's adding to the chain. And then we have rRNA, which is basically the ribosome, and that's what's creating those peptide bonds.
And somebody remind me, what changes DNA to mRNA? What's that machine? What's that machine called? I know you guys know it. What changes DNA to mRNA? What's that machine?
RNA.
RNA polymerase. RNA polymerase. And that T, think T, like what Sheila just said, transfer, right? So it's picking it up. It's transferring things. It's got that anti-codon on it, right? To specifically match to certain codons that code for certain amino acids. Picks that group of 3 up, transfers it over. Okay, and rRNA, ribosomal, pretty sure. Yeah.
RNA polymerase is what does transcription. So it's changing the DNA to the mRNA.
The protein that charges tRNAs.
You don't need to know that. You don't need to know that. I wouldn't worry. That charges tRNA? What'd she say about it?
AminoacetyltRNAs contain for each amino acid, which binds the amino acid onto the tRNA.
The anticodon? The thing that's on the bottom of the tRNA that matches?
No. Well, you're talking about what adds the amino acid onto the tRNA?
Yeah.
Yeah, I don't know specifically how that happens. I don't think you have to know much more about just like what you said, that it creates that charge that will be what adds that amino acid on top of the tRNA.
Yeah, don't worry about that.
All right. Topic 5,
When we talk about the peptide bond formation, it's tRNA is attached to the amino acid of the carboxyl, and it has the amino free. And then let's say if we're looking at the initiator tRNA, the amino is going to attack the carboxyl, right?
and then
The carboxyl of the amino acid in P site and the amino in the amino acid in A site.
okay. Topic 5, transcriptional regulation. All right, so cells want to regulate gene expression so that they can react to their surroundings. There's always stuff going on around the cells, and cells need to regulate what they're going to be expressing to respond to that. Like we talked about, transcription is just turning DNA into mRNA. And so that means that transcriptional regulation is the control of turning that DNA into the mRNA to influence what genes will then be expressed. And this is just one of the many ways that cells react to their environment and control gene expression. Some of the proteins involved, we have sequence-specific binding proteins, like the name says for those bind specifically to a certain sequence. So for that, think about things that don't want to move. They want to bind to that specific sequence and they don't want to move around at all. That would cause problems. And then we have non-sequence specific binding proteins that can bind anywhere. So for this one, think proteins that need to bind, but then we need them to move around. So if they were sequence specific, they wouldn't be able to do their job because they wouldn't be able to move to different sequences. We'll talk about that. All right, there's general transcription factors. These help RNA polymerase at different promoters. In prokaryotes, you'll see sigma factors. These bind the RNA polymerase first, and then together they go to the DNA. So they help basically that polymerase get to the DNA. Without it, it wouldn't know where to go. And then in eukaryotes, there'll be basal transcription factors, which are a little bit different because these bind the promoter first. And then once they're already on the promoter of the DNA, they recruit the polymerase. So in prokaryotes, they bind the polymerase first, then in that pair, go to the DNA. In eukaryotes, they bind the promoter on the DNA first and then tell the polymerase to come.
Does anyone know that specific one that she always talks about? She mentions on problem sets all the time. Does anyone remember that specific transcription factor that she always talks about? Anyone? That's my question.
CPK.
No. Okay, I'm gonna tell you guys right now. I swear she puts it on like every exam. She loves the specific transcription factor. So listen up. Do you remember? Sigma 70. Okay. Sigma 70. Okay. So I own a house. Okay. Like, what are some things? I'm trying to clean this house. I'm trying to upkeep this house. What are some things I gotta do? I gotta vacuum once in a while. Okay. I gotta like, I don't know, like mop. Okay. Like I need to upkeep my house. All right. I gotta change light bulbs. I gotta do some upkeeping. Just like every place I live in is gonna need upkeeping. Okay. Whether I live in an apartment or if I live in a house or whatever, I'm gonna need to do some sort of upkeeping for wherever I live. Okay. That's what Sigma 70 is. Sigma 70 is the housekeeping sigma. Okay. So things that all kind of like DNA, they kind of need. So like, okay, there's something broken, let's make sure things are working okay, checking over, seeing things are okay. They all need Sigma 70. Okay. So when we're talking about what Yago just said about sequence specific binding and non-sequence specific, I don't know if you guys remember this one question. It was like, oh, like on a scale of like not specific at all to like super specific, what do you think Sigma 70 is? So let's think about this for a second. If I have a transcription factors, remember, recruit RNA polymerase to go to a certain spot and to do things, right? At the promoter, right? That's how it knows where to go because that transcription factor is recruiting it somewhere. If Sigma 70 does jobs that like kind of need to be used in like every strand of like DNA, like basic jobs like upkeeping, do we think it's going to be more sequence specific or less sequence specific?
More?
Actually, it's going to be a little bit like kind of in the middle there, right? So I want it to be specific enough where I can like actually grab onto the promoter and I can actually keep RNA polymerase on there to have it do its thing, which is transcription, right? But I don't want it to be too specific to where I can't go and attach to like a bunch of different DNA, right? Because this is the housekeeping sigma, meaning a bunch of different DNA strands need this guy to do certain upkeeping jobs. Does that make sense? So I don't want it to be super, think of it like a spectrum. I don't want it to be super, super specific because I need it to apply to a bunch of different places that need upkeeping, but I don't want it to be too loose where, that I can't really bind to anything and RNA polymerase doesn't really care about me. Does that make sense? She's going to mention Sigma 70, promise you, some point. She does it. She loves it. Yeah. So again, for all of these questions, it's never usually really like a one or the other. I would always say like give a range. Like I would say like medium to like on the low end of high, because you want it to be there long enough for like RNA polymerase to like come in and like do its job. Like you need it to still attach, but you don't want it to be too specific to where I can't go and do it on a bunch of different DNA, because it's housekeeping. I need a lot of different places need housekeeping. Does that make sense?
I would say it's closer though to more sequence specific, because the non-sequence specific ones we talk about, you'll really know that they need to be non-sequence specific and they can go anywhere. But if it's not that it can go anywhere, think more towards sequence specific. But sequence specific, like it won't be exactly just one sequence. They'll be specific to certain sequences. That's what kind of what Anjali's talking about.
Yeah. So I would say like medium to high.
More towards sequence specific.
Yeah. But just think about that end of the spectrum. But the point is, it needs to go to different places to do jobs that a lot of people need. So it can't be super, super specific. That's the point. Does that make sense? Okay.
In general, though, those sequence 70 is always gonna bind at the promoters.
So think,
That's the sequence specific portion is that it's always gonna bind at the promoters rather than just anywhere on the DNA.
mhm.
There you go. Yeah.
So general transcription factors, like we talked about, these are just the things that are recruiting, helping the polymerase get to the promoter so it can start transcription. All right. So that's kind of why these need to be more sequence specific. They're the ones binding to that specific promoter, that specific site, telling the polymerase you can go here. So what does that kind of tell us about the polymerase? Is the polymerase going to be sequence specific or non-sequence specific if it needs somewhere to tell it where to go?
Sequence specific.
Then they wouldn't be
It would be non-sequence specific because if the polymerase was sequence specific, then it wouldn't need help getting there. Because that would be its specifics, it would be its specific sequence. So it would already know how to get there. But like we'll talk about later, it's non-sequence specific, so it needs a sequence specific thing to tell it where to go.
Which is the what? The transcription factors.
Okay. Then we have regulatory transcription factors. These regulate the rate of transcription at different promoters. We have two types, positive regulatory and negative regulatory. Like the names, positive promotes transcription. So if we have one of those, it would say transcribe that gene more. If we have a negative regulatory one, it would say stop, don't do transcription. We don't need those genes to be expressed. Does that make sense to everyone?
general transcription factors are absolutely crucial. They're needed. Can cells work without regulatory?
I would say so they can work without regulatory, but I would say most need regulatory and they just don't. If they're just regulating at regular levels, then none of them would be like activated.
Yeah.
Let's say she asks, can there be some genes or, organisms or whatever that only have general, only have regulatory? We would say no, because general is necessary.
I would think about it more like this, like cells love efficiency. We don't want everything in our cells to be turned on or turned off at the same time. So we want mechanisms to turn things on and off because that would be a waste of energy to have something always on. So by having the regulatory transcription factors, if I want more of something or if I want less of something, instead of having something completely on or completely off, I can regulate those levels. You know what I mean? So instead of just like black and white, like on or off, okay, like I need something, but I don't need like a crazy amount of something. Like we'll talk about with the lac operon. I don't need like a crazy amount of something, but I want a little bit more, right? That's how we could regulate it. Or like, I don't want to turn it off, but I want a little bit less of something. That's like how we regulate it.
So the general ones are needed. They are needed to get the polymerase to the spot that it needs to go. But these are like, they might always be there, but they're not always. It's just they're being used when the cell needs higher expression or lower expression of things. But it's not as much like the general where it's just like they always need to be there telling the polymerase where to go. These are kind of just like light switches in a house that are there and you switch them when you need them. But if not, you just leave it. But then the key would be like the general where you need to get
So
in.
general would be light switch and regulatory would be like dim.
I mean, that's good.
I was thinking more. The general would be like the key to the house. You need that to get in the house. And then the houses have light switches. You don't need to use them. You could just have the daylight or whatever. But if you want more light, you turn up on the light switch. If you want less light, turn off the light switch. All right. Consider the following and determine if they're sequence specific or non-sequence specific and why.
Let's just start with the first one, RNA polymerase. We talked about it. Sequence specific or non-sequence specific?
Well, sequence.
And why? What's the polymerase doing? It's moving, right? It starts on the promoter and then it's moving for transcription to make that mRNA. So if it's moving, why does it need to be important that it's sequence specific or non-sequence specific. Yeah, we need it to stay bound to that DNA and move throughout the entire DNA. If it was sequence specific, it would bind to a certain sequence and not be able to move. So the rest would be untranscribed. We want it to be non-sequence specific. It can bind somewhere with the help of the transcription factors and then keep moving along. Make sense?
All right, now transcription factors, sequence specific or non-sequence specific?
And why? What do they do?
Where do they bind to? Motors, right? So they're staying in place. RNA polymerase needs to go along because what does it do? It makes DNA into what? mRNA, right? It's moving. It needs to bind enough to where it can stay on, but it needs to move. Transcription factors, one place with the promoter because it needs to tell the RNA polymerase to come here. Make sense?
All right, here's what we talked about. All right, now lac operon in E. coli. So first, what operons are, they're just a set of genes that are close together in DNA and they're controlled by a single promoter. All right, so usually since these genes are all close together and controlled by a single promoter, they'll be related to each other. So they'll have similar functions. And we can either turn that whole operon off or turn the whole operon on because we either need it or we don't need those genes that are being expressed. Specifically, this operon, the lac operon, is for lactose. That's why it's called lac, right? The genes that are being expressed in this lac operon, if they're expressed, are genes that we need to digest lactose, okay? In E. coli, they prefer glucose as an energy source. So first, they're going to look to use glucose to get their ATP. If there's no glucose, then they're going to turn to lactose. And when they turn to lactose is when they need the lac operon to work, and they need these genes in the lac operon to be made so they can digest the lactose. Here you see in the picture, this is the basic structure of lactose. They can't use it like this. So we need the genes that can break it apart into glucose and galactose. You don't need to know the specifics, but just think lactose they can't use like that. That's why we have the lac operon, for it to break and be able to be turned into energy that they can use. Okay. So when, like we talked about, when there's no glucose, which is their main energy, if there's no glucose, but plenty of lactose, the cell is expressing the genes in the lac operon. What happens here is we know there's plenty of lactose. So lactose, this blue thing right here, is going to come and bind the repressor. The repressor is what was on the lac operon before, stopping the operon genes from being expressed. But now that there's lactose, it's binding to that repressor. It's taking the repressor off. As you see here, now the lactose is bound to the repressor, causes conformational shift. The repressor leaves. And now there's nothing stopping the RNA polymerase from its transcription and sliding down the DNA. So now we start to have it slide down, and it's making those genes, it's expressing the genes that we need to digest lactose, turns that into the mRNA, and the mRNA goes on, makes the proteins to digest lactose. And now we can digest that lactose that there's plenty of in the cell. If there's no lactose, then there's nothing that can bind to the repressor and take that repressor off. So the repressor would stay there, and now the polymerase is still stuck. So we're not creating genes because why do we want to waste energy making stuff to break down lactose if there's no lactose? Or if there's plenty of glucose and we can just use the glucose. We'll talk about CAP, which is a positive regulator, and that's just controlled by cyclic AMP levels. Yeah. There has to be no glucose and lactose, because lactose is important to bind to that repressor. But if there's no lactose, doesn't matter what the glucose levels are, because then there's nothing to take off that repressor. Make sense? It's a positive regulator that's controlled by cyclic AMP levels. So according to the cyclic AMP levels in the cell, if we have CAP, then since it's positive regulator, it's going to increase expression. So it's going to say we have lactose, tons of lactose, and we want to digest it all. CAP is going to work and it's going to increase expression, making it higher than usual.
And what it does is CAP assists the RNA polymerase to bind to the lac promoter. Okay, so that's how it's like positively regulating things. It's helping RNA polymerase get to that promoter.
If there's neither, there'll be little to no expression. There should be little to no expression. If there is glucose, first thing it'll want to do is just use that glucose. So if there's some, but it's only a little bit, it'll use that up and still make some of the lactose genes. So it can use the lactose too because it needs more.
And real quick, before I answer the questions, just to get the CAMP and the CAP straight, remember, everybody listen, write this down, okay? There is an inverse relationship between CAMP and glucose levels. So CAP needs to be bound to CAMP to like do its positive regulation job. How do we know how much CAMP there is? It's related to the glucose levels. All right, so you're answering these questions. You're going, number one, how much glucose do you have? And number two, how much lactose do you have? And then everything can be figured out from there. Okay, so CAMP levels rise as the glucose levels falls. Okay, and it's because like the glucose directly binds and inhibits the enzyme that makes CAMP. You don't really have to know that, but there's an inverse relationship there. CAP can only bind to DNA when bound to CAMP. Okay, CAP can only do what it wants to do, positively regulate things, if it's bound to CAMP. Okay, so when glucose levels are low, CAMP levels are going to be what? High. Inverse relationship, right? Glucose levels are low, CAMP is going to be high. What does CAP need to be bound to in order to do its job? CAMP. There's a bunch of CAMP because we don't have much glucose, which means it's going to go and positively regulate, which means we're going to get more lactose and more of those enzymes that we want to digest because there's less glucose. Does that make sense? Okay.
But I know that CAMP is like inversely related to glucose. Is it like are CAMP levels high when CAP levels are high? If that makes sense. Instead of being like inverse.
Yeah. I mean, think CAP's there when there's high CAMP, then that's when it's going to activate CAP. But the levels of CAP doesn't matter as much.
Yeah, it's there, but it can't do anything unless it has that CAMP. So CAMP is what we care about. Yeah.
Yeah, it would.
Yeah.
Yeah, that binding pocket would only bind to lactose.
Only binds.
In this operon, right? Because this operon is the lac operon and it's controlled by lactose at the end of the day.
And like, if you look at what this photo is that Yago just showed, like, I don't know if you guys remember that question that was like, oh, give it a 1, 3, 5, whatever. That's what this is. Okay, so number one, you're asking yourself, how much glucose is around? And the second thing you're asking yourself is there lactose available? Why do I care about the lactose levels? Because it's the only thing that binds to the repressor. Okay, what happens when it binds to the repressor? It's too busy being stuck to the lactose, so it's not going to repress things. Okay, so that's why we care about how much lactose there is. It's like, yes, we know glucose, like we know if there's less glucose, we should probably make some more lactose, but don't forget about the lactose levels. That's also important, okay? Because the repressor is totally dependent on the level of lactose. Does that make sense? Any more questions?
So the reason why there's a level 1 instead of level 0 when the repressor is present is because the affinity, right?
There's some leaky expression still. That's just all you have to know for that.
Right. So we have at the bottom, you can see like low level of expression, right? There's high glucose, which means we don't really need to make what we want to make because there's already glucose there, but there's lactose around. And what binds to lactose? The repressor. So that's how the leaky expression happens. And then we have the other side. We have low glucose, which means I want to make more glucose. I need energy. I need things. I need to digest. Lactose around is available, which means there's something to bind to that repressor so we can actually do it. And then we get like a whole bunch of the outcome. Yeah. Both. And if you're confused about what those numbers mean, she has it in the topic notes, like explained in depth. But I'm sure she'll give you that information as well on the exam. Yeah.
What's the last one?
The high glucose lactose available, I would say that that would probably be like a 3, right? So 3 is like there's still stuff happening. Again, notice how there's no, like, I haven't said the number 0. Okay, because leaky expression can be a thing. If there's high glucose and there's lactose unavailable, if there's no lactose, what's going to bind to the repressor? It's just going to stay repressed, right? So it doesn't matter how much glucose I have, if I have high or low glucose, if there's no lactose available, then nothing's going to bind to that repressor and then nothing's going to happen. All right, so yes, we care about the glucose because if we have low glucose, then we want to make a bunch, and if we have high, we don't really want to make much, but don't forget about that lactose. Make sense, guys? Okay, yeah.
That's the highest level you can get.
So you have low glucose, which means we need to make a bunch more stuff, and we have some lactose around, but we don't have too much. Perfect. We have something to bind to the repressor, but we also have low levels of glucose, which means we can get like a bunch more stuff made because we need it. Right? Yeah.
Should we memorize the LAC-Z, Y, and A?
You don't have to know specifically each of those, but each of these is one of those genes, and all of those play a different role in the digestion of that lactose. So it could be like Z could cleave the bond, Y could change their structure somehow, stuff like that. Just know that each of these is a different gene and they work together in the digestion of lactose. Just going over really quick these. So low glucose, high lactose. All right. Low glucose, we can't use our primary energy source. So, and there is lactose. So we want to transcribe those lactose genes. Plus, we have CAMP. We have CAMP here, the activator protein. So that's the highest level you can get. You have CAMP helping increasing the expression levels, and we have the lactose available that was binding to the repressor. So now the polymerase is like moving super fast. Think CAMP is like pushing it to make it transcribe even faster than usual.
And we have low glucose. What are the levels of the CMP? Is that going to be high or lower? High, right? Inverse relationship. Low glucose means high CMP.
All right. And then if we have high glucose, so we have our primary energy source and no lactose, we have no lactose, so nothing to bind to the repressor to take it off. But it doesn't matter because we have high glucose, which is our primary energy. So we'll just use that. We don't need to express any of these genes to digest the lactose that's not there. Same here. There is low glucose, so we would want to digest lactose, but there is none. So nothing is being expressed. And then last one, we have high glucose, have our high first source of energy, and there's also some lactose. So the lactose will take the repressor off and we'll get transcription here. But it's just low levels because there's high glucose. So our primary energy source is there. So it's not going to bring that activator protein to push it along and make higher levels of expression.
Yeah. So guys, if I was going through this, like a question, I'm looking at it and there's like glucose and I'm like, getting all mixed up in my head. Number one question, is there lactose available? Yes or no? If yes, how much glucose do we have? Then that last question is, what are the CAMP levels? Okay, because I don't care about the glucose level if there's no lactose available, because if there's no lactose available, then nothing's going to bind to that repressor, nothing's going to happen anyways. Okay, number one thing, how much lactose? If there's lactose, how much glucose is there? That's going to tell us about the CAMP levels. CAMP tells us about what? What does it bind to? CAP, right? That's why we care about CAMP. And then you have the question answered. Okay, yeah.
repressor and operator? Do we need to know that?
The LACL repressor, like we talked about, that's the repressor that the lactose binds to and can take off, or if it stays on, it stops.
Can you explain what the operator?
And the operator is just where the repressor's bound to.
Yeah, repressor binds to the operator. So she asks you a question that's like, the operator has a loss of function or like whatever. You're going in your head, you're going through the steps. Okay, operator isn't there, which means the repressor can't bind to it, which means we can't repress things, which means more stuff is going to happen. Okay, so you really have to think about it in terms of that way. Just everything is in steps, everything leads back to each other. It sounds complicated, but if you go through the steps, you know what you're doing, you know the basic principles, like what the glucose levels and the CMP levels, they're inverse, you can figure this out. Okay, do you have a question?
Are we gonna be expected to know like what lacks, dry, lactate?
I think she'll define it for you just like how she did on like the problem sets with this. Okay, let's move on. Let's just get through it for now, just because we have 2 more to go. So let's just go to the next one.
Okay, now in eukaryotes, so the operons, the lac operon is in prokaryotes, okay? That's how transcriptional regulation happens in those. In eukaryotes, it's a little bit different. Here, we have the transcription factors are binding further away from the promoter, so we need bending proteins to bring them close to the promoter to have that polymerase bind to the promoter. Because remember, like we talked about, transcription factors are what recruit the polymerase to the promoter, so it can start transcription. You can have different combinations of these transcription factors, and that's ultimately what's going to control the levels of transcription. So like we see here, here's where the transcription factors are binding to. And those transcription factors are going to recruit that polymerase to the promoter, but without this bending protein, it's going to be far away. So it wouldn't know, like it wouldn't tell the polymerase where the promoter is, right? So we need that bending protein, bring it right on top of the promoter. And now that it's on top of the promoter, it can tell the polymerase where the promoter is and put it there. And then here we have an example. So right here, we have the promoter, right? We have the transcription factors that are binding far away from it. We need these bending proteins, C and D, that are going to bend it and bring the transcription factors to the promoter so the polymerase can go. Yeah. Yeah, or in the past, she's asked questions with this. Usually she gives these images. I don't think she'll give like these words in it.
But she'll make it clear like what she means by each like shape and thing.
Does that make sense? So just like in prokaryotes, like we talked about, we have the operon being controlled by the repressor and then the camp and all those things. That's what's regulating transcription in prokaryotes. Now in eukaryotes, it's different because we have the transcription factors far away from the promoter, and we need the DNA bent so that they can be close to tell the polymerase where the promoter is.
So the biggest difference between prokaryotes and eukaryotes is transcription factors will be further away, because they need the bending proteins, and they also use the basal complex, whereas eukaryotes need, prokaryotes use the sigma 70, and their transcription factors are going to bind near the promoter.
Okay. Question. So a human liver cell and a human skin cell in the same person have the same genetic sequences, but perform very different functions, which explains why this is possible. So this goes into kind of the transcriptional regulation. B. So different genes are activated in the liver cell than in the skin cell. This is kind of talking about what we talked about we've talked about it in previous units too, but it's choosing which genes are being expressed through this transcriptional regulation. And then we have different outcomes. So all cells start with this, they have the same DNA, but we're choosing different genes being expressed through that transcriptional regulation, and that can lead to different cell types.
Does anyone remember the fancy term for that? What was that called? Differential gene expression, just meaning that we're all made out of the same DNA. The only difference between us, the reason we look different and the reason we have different traits is because different genes are expressed. Okay. So when we say activated, we mean expressed.
Okay, and then here's kind of like what we talked about, the differences in the regulation and the similarities. They both use transcription factors. Both of those transcription factors are what controls the polymerase binding. Remember, those are sequence specific. They bind to the specific sequence right near the promoter, and they tell the RNA polymerase, this is where you have to go. There's regulatory transcription factors, which who remembers what those do?
They bind to. They bind to different proteins and take them to where they need to go.
The regulatory transcription factors would bind to certain parts of the DNA, and they either upregulate or downregulate expression. So think regulatory, regulate, either positive regulatory, increase expression, or negative regulatory, and they suppress transcription. Then general transcription factors are the ones that bind to that promoter area and help the polymerase get there. Yeah. No, it would like bind to the polymerase or something and slow it down, stuff like that. Like it's not like what you're thinking where it like creates like a break in the DNA or like a obstacle. But you don't have to know like that specifically. Just know that it suppresses the rate of transcription. Okay. And then differences, we know in eukaryotes that the transcription factors bind further away from the promoter. So these are still sequence specific, but their sequence that they're specific for is just further away. And then the general transcription factors is different in prokaryotes. The sigma binds the RNA polymerase first, and then once it's bound to its buddy, it's bringing it to the DNA. But in eukaryotes, they bind the DNA first, and then they tell their friend, this is the spot where you have to bind to.
Right. All right, let's take a 5-minute break real quick, and then I'm gonna yabba you guys about 6 and 7. Okay, so we're gonna have to wake up. We're gonna have to wake up.
Sorry.
All right, everybody. It's time to wake up. It's time to wake up. I see some of y'all sleeping and snoring. Okay, we got 2 more units to get through. Okay, cell signaling. We're going a little bit more in depth with it than the other units just because it seems to be confusing people. Questions at any time, raise your hands, let me know. I'm gonna be pacing around here because I cared. Because I cared. That's my last one. Because I care. Okay. Cell signaling, topic 6. Again, any questions? Raise your hands. If you say something, I will forget what I was talking about. So just raise your hand. All right. Anytime. Okay. So, okay. That's up to I guess. All right. Basics. Cells constantly reacting to changes in their environment. So, receptor picks up signals from the outside by either binding to the receptors or taking them up on the inside, passing the message on. We're going to talk about that. The pathway we're doing talks about both, right? Signal comes in and then signal is transmitted inside. We're going to talk about all of that. All right, RTK. RTK. Remember what it stands for. Receptor tyrosine kinase. What do kinases do?
They phosphorylate.
They phosphorylate things. Phosphate. Where do we get the phosphates? ATP. ATP is adenine, triphosphate, whatever. That's where they get the phosphates from. Point is, kinases phosphorylate things. Why would we want to phosphorylate something? Why do we care?
Give it energy.
Your energy, it can kind of be used as like an on and off switch. Okay, so instead of creating an entire new protein from scratch to do a certain thing, I can turn on and off things through phosphorylation. This is just one of many ways we can turn things on and off. But when we're talking about the term switch mechanism, if she ever mentions that term, switch mechanism is like the on and off switch, okay? So how do we do the switch mechanism? We can phosphorylate something. We can add a phosphate. We can take it away. That's going to turn it on, then off, okay? So these are little ways that we can, mess with proteins that already exist and have them do things that we want without having to make an entirely new protein. Okay, so the phosphorylation is important. So let's talk about the beginning. Okay, this is the RTK. Tyrosine kinase. You guys remember tyrosine?
Yeah.
Little amino acid. That's important. Okay, we're going to talk about the 2 different types, but there's tyrosine kinase domains. Okay, so this is the RTK. They're inactive. Notice how they're 2 separate parts, right? They're 2 separate parts. They're not together yet. Tyrosine kinase domain. Okay, that's here and that's here. That's important. We'll talk about that in a second. So first step, signal comes in, binds to the receptor. When that happens, they I think it's dimerize or demerize. I was wrong. Dimerize. I was going to say dimerize. Dimerize means 2 units become 1. Okay, they dimerize, they come together. When they come together, they cross phosphorylate, which means they phosphorylate each other. Right? Phosphorylation means what? We're adding a phosphate onto something, right? In this case, we're using it to turn it on. Okay, so signal comes in, dimerization. Then now these 2, the receptor kinases, so these 2 guys are now active and they can cross phosphorylate each other. Okay, so that's the first part. Quick question. Before we even get into the pathway. Talk about phosphorylation. We know that TK1, TK2, we're gonna get into all that. They get phosphorylated. Are the phosphates passed from RTK, TK1, TK2, TFA? Is it like a relay baton?
Yeah.
And if no, why? Answers, yeah. Okay, yeah, let's talk about eukaryotes, right? I'm gonna say no. I'm gonna tell you why. So each phosphate is placed on a protein by a kinase that are taken from ATP. So we're not passing the same phosphate from one to the next to the next. We're not doing that. Every single time we phosphate something by the kinases, they're taking it off the ATP and they're attaching it to something, okay? So when we talk about, oh, and then this gets phosphorylated and then this gets phosphorylated, when we talk about that part of the pathway, these are all different phosphates. Okay, so they're not being passed from each step to each step. Each kinase is taking a phosphate from ATP and placing it on a protein. Does that make sense? Okay. Let's get into the pathway. So we went over the RTK. First step is what? Signal comes in. Next step is what? What is that term I just said? That
Dimerization.
dimerization. 2 things become 1. When 2 things become 1, they can cross phosphorylate each other Those little circles in the middle there, they're active. They can do that now. Okay, now, so RTK, we don't really know how. It somehow activates TK1. Okay, so big rectangle at the top. RTK can activate TK1. How? We don't know how. Okay, TK1 can now phosphorylate TK2. Okay, and then the last step here is TK2 can phosphorylate this TFA. And once that happens, TFA can enter the nucleus, bind to this, and alter transcription. Okay, so I'm not just passing a signal through. I'm starting a signal that's going to turn on something, it's going to turn on something else, it's going to turn on something else. But all I really care about, guys, I don't care about any of these guys in the pathway other than this guy. So like, let's say everything else in this pathway was inactive. TK1's inactive, RTK is inactive, TK2 is inactive. As long as this guy is turned on, which we'll talk about how that can happen, then I get outcome, right? So yes, I would love for all of these to activate TFA, but it is possible for it to be turned on. We'll talk about how that happens in a bit, and then give me my outcome, right? So when you're talking about things that are being messed with in this pathway, she's going to give you a lot of those questions. She's going to say, if TK1 is gone and you just have TK2, but TK2 is still turned on, am I still going to get an outcome? If TK2 is still turned on?
No.
Yes, because if TK2 can phosphorylate TFA, then I'm going to get my outcome. And we'll talk about how that is even possible. All right, but just remember, TFA is what I really care about.
Okay, so think TKs, all those, the Ks, kinases. So those are all phosphorylating the next step to activate. The TF is transcription factor. And like what we talked about, that transcription factor, when it's activated, is going to go into the nucleus, bind to that DNA, and cause the transcription to happen. Okay, and then like she was saying, how it's not like passing a baton. Each of these kinases, think of it more like a relay race. So the start one is activated, goes to the next one, tags them, they're in. Now the next one's active, right? A baton would be like you have the baton, you pass it, now you don't have it anymore. So that'd be like you have the phosphate and you're passing your phosphate. That's not what's happening. They have their phosphate, they keep their phosphate, and they're putting a different phosphate onto the next one, activating it. Does that make sense? The next,
Whatever the next thing is.
So then the TFs are just like waiting to be phosphorylated.
Yeah.
Exactly, they're waiting to be activated by TK2 with the
So
phosphate.
matter one TK isn't phosphorylated as long as like one of them.
We'll
as
talk about that in a second.
as long as the last one is active, everything before could be off.
Okay, so quick thing about kinases. Kinases phosphorylate to either activate or deactivate. It's not always going to be activate. It's not always going to be deactivate. But the point is, it's a switch mechanism, right? We can use it to turn things on or off. Takes phosphates from the ATP, turns into ADP, whatever. Don't really have to know this. Just know that again, when we're not passing the phosphate along. Each time we activate something with the phosphate, we're taking it off the ATP and we're putting it on the protein. Got it? Yeah.
Receptors, are there different kinds of receptors that take into all types of signals? Is it one kind of receptor on that side? Sorry.
Yeah, no, for different pathways, there can be different types of receptors. So what's special about this one is there's tyrosine, right? Tyrosine receptors can only really interact with another tyrosine receptor, which is why they come together and cross phosphorylate. So yeah.
You don't specifically know. She'll usually say it. Most of the times it's activating, but there is some times where it's deactivating. But she'll say that if you need to know it.
We know because scientists have like studied this pathway, and so they think that the phosphate turns something on, but that's not the case for all pathways.
That last part.
Oh yeah, phosphate can be removed by what we call phosphatase. Does anyone know what it means when we have ASE at the end of something? What does that mean? It's an enzyme, right? So this is an enzyme whose only job is to remove phosphate. So like what we said, we're not passing the phosphate from one guy to one guy to one guy. We're assuming that they all keep them. but we don't want them all to be on forever. So what takes them off? Phosphatase, right?
And phosphate group isn't just added to any part of that protein. It's added to specific hydroxyl parts of the side chains. So what that means, hydroxyl is just an OH group, okay? And there's only certain amino acids that have those hydroxyl groups on their side chains. So those are the only amino acids that can be phosphorylated.
But don't get too wrapped up in the kinases. Just know that kinases phosphorylate things, and in this case, the pathway that we're talking about is turning them on.
And that phosphate group changes, either turns something on or off because it changes the structure, right?
And structure determines what? Function,
Function.
right? Okay, great. So we just talked about this. I'm just going to speed through it because we got a lot to get through. Okay, question. If analysis of tyrosine kinases in a cell shows that RTK is not phosphorylated, RTK is those two guys up there, right? But TK1 is phosphorylated, will the pathway outcome still be produced, and how could this be possible?
Yeah.
How could this be? Okay, let's do outcome, yes or no?
Yeah.
Yes. How is this possible? How can we have one thing turned on, another thing turned? Yeah. Crosstalk. All right, we're going to talk about that in a second, but that's the answer, but we'll come back to that. Okay, 2. If TK2 is missing in the pathway, would the outcome still be produced in the presence of a signal? No. No. But asterisk, 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 asterisk. That's what we're going to talk about in a second. There is always, always, always the possibility of crosstalk. Okay, and we're going to get into that in a second. Yeah. Sorry, pick it one. Okay, so before we get into this, real quick, can we go to the signal amplification and the crosstalk? Yeah, it's good. Let's get there.
Bit more.
Yeah. Real quick. Yeah, there it is. Okay, signal amplification. Let's get into this first before we even talk about crosstalk. Signal amplification. If I tell Ty a secret and I'm like, Ty, don't tell anyone, but because Ty is sneaky and he's going to tell everybody, he's going to tell all his friends. And then all of his friends are going to tell all of their friends. And now my secret is out. Now my secret is spread. So it's like gossip. Okay, that's what signal amplification is. All right. We start with one signal to one receptor. That receptor goes to a bunch of different people. Each of those go to a bunch of different people. And then you have a message being spread. Okay, that's how signal gets spread. Does that make sense?
Yeah.
Right? Okay, so signal amplification. If you notice here, there's like what, like 1, 2, like 3 steps. Okay. I can have a pathway with more steps, and I can have a pathway with less steps. So I'm telling Ty my secret, and he goes and tells like 2 other people, and they go and tell everybody they know, and it stops there because they don't really know anyone else because they're losers. I don't know. Okay. What is a pro of having a shorter step pathways or less steps in a pathway? What's a pro? Yeah. More efficient. Why? Because there's less steps happening. There's less people I got to go talk to, which means my message is going to be spread faster. Right? If I'm trying to get to you and I tell Ty something, he only has to go back 1 and tell you, right? But if I want to get it all the way in the back and I have to tell Ty and then Ty tell her, then, you know, it's going to take a lot more time. So that's pro number 1 is it's going to be a lot faster to get a signal across 5 less steps in the pathway. What's a con of having less steps in the pathway? It's a con. Yeah. Yes, less opportunity for signal amplification, meaning my message is not going to be spread as far. Makes sense. Okay. And now what's a pro of having more steps in the pathway? What's a pro? We know it's going to be slower, right? Because I have to get through so many more people just to get to the back or wherever I want to go. What is what's a pro of having more steps in the pathway? Anyone know this? No, you know it. Anyone else? We mentioned the term earlier. What's that term I haven't talked about yet? More opportunities for crosstalk. And let's get into crosstalk. Crosstalk. One linear step pathway is talking to another linear step pathway. Okay. So let's say, there's pathways for like survival signals. I need survival signals for the cell to survive, okay? Let's say there's no survival signals going on. Another pathway can come in and be like, hey, get that going. Like that cell actually needs to survive. And I can turn on something in that pathway. That's crosstalk, okay? So it's different pathways talking to each other. That's like what this big thing is. Don't worry about any of the names here. I don't even know what this means. It's just like two different pathways talking to each other. It's like being in two group chats and texting one according to what the other says, okay? So going back to that question earlier of, if you can't get to, that's cool. Almost there. While he's doing that, I'll just say, how is it possible that a signal comes in, the RTK isn't working, but I can have TK1 be phosphorylated? Crosstalk. Okay, so that means another pathway can come in and turn on TK1. Doesn't matter that RTK isn't activated, but another pathway can come in and do that. Okay, it also means another pathway can come in and turn something off. All right, so even though you're answering number 2, if TK2 is missing in the pathway, would the outcome still be reduced in the presence of signal? The answer is no, right? Because if this is missing, then who's going to activate this? But the asterisk here, and this is what you're going to write in your exam, but there is always the possibility of crosstalk. Okay, so there's always a possibility that another pathway could come in and activate this guy, and the outcome could still happen. Okay, so assume when you're answering these questions that there's no crosstalk happening, but it never hurts to mention. Okay. Because you never want to say never in bio. So your answer is going to be no, but asterisk, there's always opportunity for crosstalk. Does that make sense to everybody? Crosstalk, signal amplification. Any more questions on that? Okay. All right, now let's talk about modulating signal pathways. So once a cell receives and outputs a signal, the cell needs to modulate the set output signal. So what does this mean? We need to regulate how strong the signal is. Okay. So what are things we can do to change that? The number of components in a pathway can determine the strength of a signal, right? So we're talking about more steps, less steps. If there's more components in a pathway, there's more steps that need to be done, that's going to amplify our signal, right? Because it's going to be spread to more places. If I have less components in a pathway, that signal amplification is going to be less, right? Toggling on and off the components of a signal. How do we do that in this pathway? What do we say? What was that term that we do? We phosphorylate, right? That's how we can turn on and off things, right? That's that switch mechanism. And so here are the examples that we're going to go over. We're going to go over feedback loop, ubiquitin mediated proteolysis, whatever, and receptor mediated endocytosis. We're going to get to each of them. Feedback loops. Feedback loops, listen to me for a second. Listen to me. All it means is that something downstream, something later is affecting something earlier. That's all it is. That's what a feedback loop is. Positive feedback loop means you're creating more of something, right? You're having more of a response because of the feedback loop. Negative means it tells something upstream to decrease something. Okay, so that's positive and negative. Positive, more, pretty easy to remember. Negative, less. Got it? Oh yeah, then that's what I just said. Okay, receptor-mediated endocytosis. When the cell doesn't want any more signals to be picked up by the receptor can actually be brought into the cell through endocytosis, creating a vesicle. Okay, and that vesicle is sent to the lysosome to be degraded. So we had receptor out here, it gets brought in, creates a vesicle, and that vesicle is sent to the lysosome. I'm going to say the proteasome, right?
Lysosome in this case.
Lysosome in this case. We'll get into the other one later. But basically, receptor, sucked in, vesicle, sent to the lysosome to be degraded. Okay, let's get into the next one. Ubiquitin. Okay, this is a little bit different. Instead of the receptor just coming in, vesicle sent to the lysosome, we have a different system. Instead, I'm going to tag the things with ubiquitin that I want to be degraded. So I tag it first. I'm telling the cell, hey, I don't like this guy. He's got to get out of here. I'm going to tag him with ubiquitin. Okay, those tags will be targeted for destruction. Okay, so anything tagged with ubiquitin is going to get sent to the proteasome. Proteasome is the garbage disposal of the cell. Okay, proteasome, garbage disposal of the cell. Anything gets sent there, it's degraded. Okay, so the difference here is receptor is being sucked in through a vesicle, sent to the lysosome. This one, it's getting tagged, any protein getting tagged and sent to the proteasome. Proteasome is the what of the cell. Proteasome is the what of the cell. The garbage disposal. Okay, yeah.
Is it tag proteins that like are not working?
Yeah, not working. It can do it for like a whole bunch of reasons. But again, remember, cells love efficiency. So if something's not working or something isn't performing up to par, we want to get rid of it. Yes.
I know. What that.
What's actually telling ubiquitin to get rid of something? It's like a ligase system. It's like they basically like pass it on to each other, and then that last guy like tags something. But there's basically like this like alarm response that happens. It's like something's not working or something isn't performing the way it should, and the cell has its own way of dealing with that. But
And
you know,
the ubiquitin is probably just like a signal sequence that tells it to end up at the proteasome.
okay, talk about simplification. Crosstalk. Okay. Are there more opportunities for crosstalk or simplification in a 2-step pathway or a 10-step pathway, and why? Which one has more opportunities? 10-step. Somebody tell me why. Somebody tell me why. No way. Come on, guys. Yeah. More components, more steps, more opportunity, more people to talk to, more people to spread it to, more, it's taking longer, so there's more opportunity for another linear pathway to come in and do crosstalk, turn something on, turn something off, do whatever it needs to do. All right. We made it, guys. We made it. We are at topic 7. We should all be proud. We should all be happy. Okay, yeah, little round of applause, maybe a little round of applause. We made it. We made it. We're here. We're here. Okay, cell cycle regulation. Let's get into it. So, what does G stand for? Growth. G.
Oh, yeah, growth. Oh,
G, guys.
Oh, growth.
G. G stands for growth. Okay. G0. Cell is doing its job, uninterested in dividing. It's low-key just chilling. It's chilling, it's in G0. It's chilling, it's growing, it's doing its thing. G1. Beginning of cell cycle ends with the restriction checkpoint. Point of no return. What do I mean by this? What do I mean by the restriction checkpoint? Yes. Yes. So until a cell, growing cell, until it gets to the restriction checkpoint, it's got some commitment issues. It's like that doesn't really know. It's like a little situationship with the cell right now. We don't really know if it wants to commit. Gets to the restriction checkpoint. If it gets through, we're doing this. We're doing the thing. We're going into S phase. We're doing replication. We're doing the whole thing. S phase, synthesis, replication, DNA, right? G2, G stands for growth, more growth, and the final prep for the M phase. M phase is where chromosomes are divided in new cells. Okay, but restriction point, important. So if I ask you, when does the restriction checkpoint happen? What are you going to tell me?
G1.
When in G1? Late G1, right? So whenever she asks you to name something or like name when a specific thing happens, don't just say the phase. Make sure to always say like exactly where you think it is, right? Because those answers can differ.
And then if you hear interphase is everything except M phase. So M phase is when it's actually dividing. Interphase is everything else other than that. So G1, S, and G2.
Okay. All right. Here's how you're going to remember this. PMAT. Prophase, metaphase, anaphase, telophase, and then synaphases. Okay, PMAT. All right, prophase. Chromosomes condense and nucleotide break apart. Spindles begin to form. We're just starting. Metaphase. Chromosomes line up in the middle, right? I think M, metaphase. I think middle. Chromosomes lining up in the middle. Okay. Anaphase. Chromosomes begin to be pulled apart. A apart. Anaphase. A apart, right? So we had metaphase. M is for what? Middle. And then anaphase, A is for apart. Okay. Telophase, new nucleotide form. New nuclei form, and membrane begins to pinch. Okay. So it does that little, yeah, little great visual demonstration here. And then cytokinesis is when those two cells completely separate. They're formed. Okay, pretty straight up. PMAT. Again, remember M stands for what? Middle. A stands for parts. T. I don't really have one for that one. If anyone does. What is it? Two. kind of like we're beginning, right? Two nuclei, kind of. And then cytokinesis. That's just like the one on the end. Okay, CDK. What do kinases do? Phosphorylate stuff. All right. And it's dependent on cyclin. Oh, how convenient. It's in the name. That's so great. So if you ever forget what it is, remember what it stands for. CDK is the main cell cycle control too. So it's controlled by cyclin. Remember, it's cyclin dependent. Cyclin phosphorylation, dephosphorylation, and by CDK inhibitors. And it requires cyclin dependent, whatever. Okay. So I have a bunch of M cyclin coming in. I have the CDK1. I have an inactive M CDK. Okay, M is for what? What phase is that? Mitosis. Yeah, that stuff. Inactive M CDK. And it comes in two parts. Can you go to the next one? Okay, right. So this is important. Okay, wake up. This is important. So there's two parts to this. Okay, it adds it's inhibiting phosphate to CDK. Remember when I said that when we add phosphate stuff, it's not always going to turn it on? This is an example of that. Okay, WEE1 adds an inhibiting phosphate to CDK. Inhibiting means like stops, right? Inhibiting. CDC25 removes this inhibitory phosphate and activates CDK. What do we call those things that remove phosphates? Phosphate. Yes. Right? So CDC25 removes this inhibitory phosphate and activates CDK, which means CAC phosphorylates. Does anyone know what CAC stands for? What does CAC stand for? Somebody say it louder.
CDK activating kinase.
CDK activating kinase. How convenient that it's in the name, right? So CAC comes in, CDK activating kinase. It's already telling us that when we're going to add a phosphate to something, we're going to activate it. It's in the name. That's great. It's going to phosphorylate it and activate CDK. Okay, so remember, step one, WEE1 adds an inhibitory phosphate, meaning we're blocking stuff from happening by adding this phosphate. So like the pathway we talked about before. Second thing is CDC25, a phosphatase, removes that and activates CDK. Okay, so that's activation one. Notice, activate CDK, CAC phosphorylates and activates CDK. So it's not just one activation, CDK, it's two. Why is it two? Because this guy, CDK is so important. It's so important. We need CDK. It's like you gotta bring in the big guns. Okay, so I have two different activations. So what's step one? WEE1 does what? Adds a what? Inhibitory phosphate. Step two, the phosphatase CDC25 gets rid of that guy. That's the first activation, activation one. And what's activation two? CAC phosphorylating and activating CDK. Okay, so there's activation one, there's activation two. Remember that. It's not just super easy. And remember, adding a phosphate to something doesn't mean that you can always turn on. And in this case, it's like turning something off. Right? So to get rid of that, we need that phosphatase CDC25 to come in, take it off. Activation one, activation two, CAC, CDK activating kinase comes in, phosphorylates it, activates it fully. Got it? So if I have one of these, is it going to be activated? If I have one,
No.
no. If I just take it off, is it going to be activated? No. I need both. Got it?
So CDK's active state is only with CAC and CDC25 and without WEE1.
Yes. So WEE1 comes in, puts that inhibitory one on. We need to get rid of that for it to even be activated. So that's step one for the first activation. And then we need that CAC to come in and finish the job. Okay. Okay, cyclins. Cyclins must bind to CDK for cell cycle regulation. Remember, CDK is the main cell cycle regulator. Okay, it's the big guns, the mafia boss. Okay, it's the guy. Cyclin levels change throughout the cycle and according to when they are needed. What a convenient system, right? So CDK doesn't know what to do until the cyclin is set, right? Because it's cyclin dependent. So you can see there's a lot more G1 cyclin, spikes here as cyclin, like it corresponds to each one and it goes down at a certain point because CDK is cyclin. Okay. Yeah.
No, just the cyclins usually have the letter name because they're associated with that phase, usually, and the same with the CDKs, but you don't need to know specifically.
I have a feeling that the main thing she's gonna ask you about is what the We1 CDC labs. Okay, G1, restriction checkpoint. Cells don't want to commit to the cell cycle and replicate their DNA if they're not ready. It's that commitment issues, okay? Cyclin binds CDK, cyclin CDK phosphorylates RB. So remember, so these guys right here, yeah, whatever, RB, okay? RB releases E2F, this little guy right here. And once that happens, E2F enters the nucleus and cell moves into S phase. So when they're bound together, is the cell going to commit to the cell cycle? No. E2F wants to be free. He wants to be free so bad. Okay. And then E2F itself literally enters the nucleus and the cell moves into S phase. Okay. So they need to come apart for the cell to commit to the cell cycle. And how does that happen? Growth factors, blah, 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 and cause the increase in cyclin and E2F concentrations. Cyclin binds to CDK. Up there, CDK is cyclin dependent, right? CDK is phosphorylated, that whole thing happens, right, with the We1 and the blah, blah, blah. RB inactivates E2F by binding to it. So when they're together, things don't happen. When they're apart, things happen, okay? CDK is activated by dephosphorylation, and it catalyzes the phosphorylation of RB. RB releases that guy, it enters the nucleus, and it starts S phase. Okay, so those two parts come together. We just talked about the whole activation of CDK. This whole guy right here, okay, cyclin dependent, connected to RB and E2F. Once CDK is activated, it phosphorylates RB. Okay, so that's the last step. When RB is phosphorylated, it can let go of E2F. If RB is not phosphorylated, is it going to stay together or come apart? It's going to stay together, okay? So RB needs that phosphorylation to let go of E2F, and once that happens, we can go into S phase, okay? All right, spindle assembly. She also loves spindle assembly checkpoint. Cells don't want their chromatids to separate until they are connected to the spindle. Securant, secures, separates, inactive. Okay, so what does all this mean? Inhibitory protein. That's securant. That brown guy up there. Okay. This enzyme over here is separase. Okay, separase separates the cohesions, holding together the chromatids. Okay, which kind of makes sense. Separase, securant, it's securing separase. So it's like not active. It's kind of like the same thing. So APC, this is APC right here, ubiquinates securant, the brown guy up there, to release this guy. So kind of same thing with RB and E2F. It's just a different method. Instead of phosphorylating something, we're ubiquinating it. Okay, so we're ubiquinating the brown guy, releases separase, and once that happens, this is the before and this is the after. We can pull stuff. Okay. Yeah.
But activate frequency decides whether frequency to
You don't need to know that. You just need to know that when it's active, it's what's going to ubiquitinate the securant. Same thing. The spindles are just made up of microtubules. So like the spindle would be all these are spindles, and they're made up of microtubules. Makes sense?
So step one, APC is active. You don't really know how it just gets active. It just is. APC comes in, ubiquinates securant, the brown guy. When that is ubiquinated, it releases separase, which separates things. And then it can pass the spindle assembly check. Okay. Okay, P53. Again, she loves P53. She loves P53. Okay. Two terms you need to know. P53, P21. She's going to have those on there guaranteed probably. P53 is activated during DNA damage. So it's part of the cell's like alarm response. Okay. So if the cell says something is going wrong, DNA damage is happening, we don't want that. P53 comes in. The only job of P53 is to activate P21. P21 is a CDK inhibitor. CDK needs to be activated for cells to move on, right? It's the main cell cycle regulator. We need that guy. So if we're inhibiting it, that's not good, right? CDK inhibition pauses the cell cycle to allow for DNA repair. So this is really cool. So something's going on. There's sense of DNA damage. Step one, is we're gonna upregulate the transcription of P53. P53 is gonna activate P21, which is gonna say to the CDK, hold on, we need to check some stuff out, we need to fix some stuff. And then that's what's gonna allow us to pause and allow for DNA repair. Okay, if it cannot be repaired, it'll just die. Apoptosis, cell will die. Yeah.
Wait, so P53 doesn't repair the damage P21?
Actually, none of them do. So P53 is up here. P53 just tells P21 like, hey, you gotta go and stop CDK because there's cell damage going on here. It doesn't actually like repair it itself. It just stops things. It's just for this pause response, the break response when things are going wrong. Same with P21.
Oh, it's like P21.
Yeah.
Stop,
Yeah,
They
so
stop the cell cycle.
so remember CDK, if it's active, if CDK is active, it tells the cell to keep going, to keep dividing, right? If there's damage in the DNA, we don't want to divide because then that'll just make cancer. So in this case, if we detect damage in the DNA, P53 is alerted. P53 tells P21. P21 tells CDK stop. And so now CDK isn't going and continuing the cell cycle. It's just stopping and there's nothing to continue the cell cycle. So it pauses there. And then it's going to pause like that until it can fix the DNA. And then it'll reactivate the CDK, or if it can't be fixed, it'll just kill the cell.
So CDK, if we're talking about like, we want to press on the gas, right? We want to make and differentiate a bunch of cells. The gas is like CDK is working. Okay, just keep going through the cell cycle. We want to go on the brakes a bit. We want to hit the brakes. That's P53. But remember, they're not actually doing the repairing. P53 is all it does is tell P21 to go and stop CDK. So we can pause and we can assess the cell damage. Got it? Remember those two. She'll definitely ask about them. Okay, cancer, not fun. Okay, when cells uncontrollably divide, right? That's at its core. That's just what it is. So she's going to ask you for two different ones. I'm sure she's already asked you in your problem sets. Proto-oncogene, continuous cell cycle and can cause cancer. So let's say if the break response didn't work well or something's wrong with P53 or P21 and it just keeps going, proto-oncogene, okay? So things are bad, but we're still going for some reason. Maybe something's messed up in this like alarm response. Tumor suppressor genes, stop cell cycle, stop cancers. And then a couple more terms. GOF mutation means gain of function, means it's overactive, right? Maybe that's a bad thing, maybe that's a good thing. Loss of function mutation, LOF, underactive. Any questions about these terms? Yeah.
So the way that is it worded, does that mean it technically still can be in some cases? Loss of function, like it says underactive, so that means it just only sometimes functions or never?
It suppresses the function. So it can suppress it just a little bit more than normally, or it can completely turn it off.
Okay, got it.
Yeah.
So for cancer, we would say that a loss of function in tumor suppressor and gain of function in proto,
Exactly.
always.
Well, or it could just be, it doesn't have to have both. It could be a loss of function in a suppressor. And
Depends
that,
on what's causing it. So we'll talk about some examples in a second. So, real quick, what are 4 ways CDK is regulated within cells in order to regulate the cell cycle? What can tell CDK what to do or what not to? What's one thing? What's in the name? What's in the name?
Like
Cyclin. Levels of cyclin. What's another thing?
RV,
RB. I'm thinking think more simple. Like a CDK what? Inhibitor, right? What are 2 other ways?
and then one last one.
One more.
dephosphorylation.
Well, that's with phosphorylation.
Yeah, that'll be in one.
But what can we do to CDK when we don't want it anymore?
Oh, ubiquitin.
Ubiquitin, right? Or just levels of CDK. So if the cell's not expressing high levels of CDK, then there's not enough CDK, then it regulates it that way.
So in the name, cyclin, right? It's dependent on cyclin, depends on the inhibitors, right? Which straight up go like P21 straight up go and inhibit it. phosphorylation, dephosphorylation, right? Like We1, putting the inhibitory phosphate, right? Those are all things. And then the levels of CDK itself. So deleting components, adding, like, those are all ways that we can control a response. Does that make sense? All right. Which are proto-oncogenes and which are tumor suppressors and why? Remind me which one proto-oncogenes is. Can somebody define that for me real quick? What does that mean?
It continues the cell cycle.
Continues the cell cycle, right? Which means cancer if there's damage, right? And then tumor suppressors, suppressing tumors. It's the opposite, right? So let's start with the first one. Which are proto-oncogenes? Which are tumor? Let's just start with proto. Which ones do we think are proto-oncogenes?
Yeah, two and stuff, and like.
Just give me one. Just give me one. Somebody raise your hand and give me one. Yeah. P53, why? Okay, it stops it. So which one are you saying it is? Are you saying it's proto-oncogene or are you saying it's a tumor suppressor? Tumor suppressor. Right. Okay. E2F, proto-oncogene or tumor suppressor? Proto-oncogene. Okay. What do we know that E2F does? What does it do? Yeah, it commits cells to the cell cycle, right? Once it's released, we can go and commit cells, which means we're going to keep going with things. Okay, separase. Separase, remember, separates at the spindle assembly checkpoint. Proto-oncogene or tumor suppressor? Proto-oncogene. Why? Because that's the only way we can get through the spindle checkpoint is if separase can go and do its job, released by securin to pull those through. Yeah. Yeah, right? Because if it's still binded to separase, then separase can't be free and separase can't do this, right? Which means we don't pass which checkpoint? Which one? Spindle assembly checkpoint. So there's that first checkpoint we talked about, which is the what checkpoint? Restriction, which is in where when is that? Late G1. And when is the spindle assembly checkpoint? Somebody tell me. Okay, all right. And CDK. CDK, proto-oncogene or tumor suppressor? Main cell cycle regulator.
Okay, so like we talked about, P53, when P53 is active, there's DNA damage going around. We don't want the cell to continue dividing, so it tells eventually P21 to tell CDK inhibitor to work. So CDK would stop, meaning it's a tumor suppressor. E2F is what is being held on to RB. When E2F is free, it tells the cell to continue. So proto-oncogene. Separase is what separates the chromatids together. When it's working, it allows the separation to happen and allows the cell cycle to continue. So that's why it's a proto-oncogene. And CDK always activates this next step in the cell cycle when it's active. So that's why it's a proto-oncogene. If there's a gain of function in a tumor suppressor. So if there's a gain of function in a tumor suppressor is stopping division. A gain of function would mean that it's always stopping it. Yeah, basically, it would just constantly be stopping it.
Yeah. The question she'll give you will be like, if there's a gain in function in like securin, what is that going to do? Proto-oncogene tumor suppressor. Securin holds on to separase, right? And so if it has a gain of function, if it's doing that even more, I mean, separase isn't going to be, you know what I mean? That's how you have to think about these questions.
When you think of loss of function and gain of function, think what its job is, like what its main job is. Gain of function would be it's doing that job more. Loss of function is it's not doing its job well. Yeah, so a proto-oncogene would be enhancing cell division or would be causing it to go forward. If there's a loss of function, if its main job is to go forward, now it's not doing its job as well, so it's not pushing it forward as well. It can be a good thing, but proto-oncogenes don't always cause cancer. It's just like if there's cell damage, sometimes we need things like we need CDK. It's a proto-oncogene, but we need it to activate the parts of the cell cycle as long as it's ready.
Okay, and that is it. Thanks for coming, guys.
Thank you.
Really appreciate it. We recorded it. If it saves correctly, we'll send out the recording.